Hello everyone. In this lecture, we are going to discuss about one of the topics from medical microbiology, that is tuberculosis. So, the causal organism of tuberculosis is Mycobacterium tuberculosis. This Mycobacterium tuberculosis primarily affects the lungs if the infection gets untreated. So, there are two types of tuberculosis. One is pulmonary tuberculosis, which affects the lungs, and the second one is extrapulmonary tuberculosis in immune weak patients. So this extrapulmonary tuberculosis is actually the infection spread out of the lungs. Means if there are some regions which actually infected by the tuberculosis apart from lungs. So this extrapulmonary tuberculosis occurs in immune weak patients. And the main cause of tuberculosis will be it's an airborne disease, which means it's transmitted through air. Epidemiology of tuberculosis. About one-third of the world's population is infected with Mycobacterium tuberculosis. So this epidemiology shows about the regions or geographical areas in which the infection had spread and about the count of the people. So about one-third of world population is infected with Mycobacterium tuberculosis, and each year 3 million people dies with this infection and 8 million new cases of tuberculosis occurs. So the problem is much larger in Africa and Asian regions, and this tuberculosis is 40 times more common in HIV patients, HIV positive patients, than in the general population. So if you observe the patients who are infected with human immunodeficiency virus or who are suffering with AIDS, they generally have a weaker immune system. So as I have mentioned that the person with weaker immune system had more adverse effect of tuberculosis, and here they mentioned 40 times more common in the patients of HIV than in the general population. So pathogenesis. Pathogenesis show the pathway how the microbial organism enters into the body and how it spread the infection. So this Mycobacterium tuberculosis enters the body and primarily infects the alveolar air sacs which are present in the lungs. So when you observe the structure of lungs, so lung consists of bronchi and bronchioles, and at the end of bronchioles we have a sac-like structures known as alveolar air sacs. So this is where the exchange of gases occurs. So this Mycobacterium tuberculosis, upon its entry into the alveolar air sacs, this microbe invade and multiply in number and replicate within the endosome of alveolar macrophages. So the macrophages are the immune cells. So the macrophages are the immune cells which actually present in each and every organ so that they can protect the particular organ. So if you see the macrophages in the liver are known as Kupffer cells, and if it in case of lungs, those are known as alveolar macrophages. So this Mycobacterium tuberculosis, upon its entry, invade the alveolar air sacs and multiply its number and replicate within the endosomes of alveolar macrophages. Because the function of alveolar macrophages is to detect tuberculosis and try to kill it. So in order to abolish this kind of immune cell, the mycobacterium actually replicates within the alveolar macrophage. So in order to protect themselves from the phagocytosis process. And so the main activity of the alveolar macrophage is whenever they find an antigen. See, for example, these are the antigens. So this is the alveolar macrophage. So whenever they find a particular antigen in the lungs, they engulf this antigen and forms phagolysosomes in its body, and it actually digests the particular antigen. So by this phagocytosis process, they could be able to protect our body. So in order to prevent this defensive mechanism, so this mycobacterium targets the alveolar macrophage and replicates within the endosome so that it could be able to protect itself from this phagocytosis process. So, however, the Mycobacterium tuberculosis has waxy mycolic acid capsule that protect it from these toxic substances. So, even though these immune cells may secrete some toxic substances in order to destroy the Mycobacterium tuberculosis, due to the presence of a waxy mycolic acid capsule on its surface, it protects itself from these toxic substances. So Mycobacterium tuberculosis is able to reproduce inside the macrophage and will eventually kill the immune cell. So as I have mentioned, as alveolar macrophages are the protectors of the lungs, the Mycobacterium targets the protectors and replicate inside the cell and destruct it, then only it could be able to spread the infection. So, due to the presence of its mycolic acid coat, also, whatever the substances are there to kill the mycobacterium, they won't be able to kill that microorganisms due to the presence of this waxy mycolic acid capsule. So, this is how the mycobacterium survive. And after the killing of immune cell, they spread the infection, causes tuberculosis. And tuberculosis is classified as one of the granulomatous inflammatory disease. So this is very, very important here. So granulomatous inflammatory disease. So this mycobacterium, upon infecting the lungs, there will be a formation of a structure known as granuloma. Granuloma. So upon the formation of granuloma, also the mycobacterium will survive and cause inflammatory diseases. So let's see the mechanism. So what is granuloma here? So the granuloma, formation of granulomas, which are organized collections of immune cells, primarily macrophages, that form in response to chronic infection with Mycobacterium tuberculosis. So the granulomas are essentially the body's attempt to contain and control the infection, but they can also contribute to tissue damage and disease progression. So this is how the granuloma forms. In order to form granuloma, first of all, wherever the antigens are settled, wherever the Mycobacterium tuberculosis settle, at that particular spot, the macrophages comes together, form an association. So this macrophages, not only macrophages, so these macrophages are basically giant cells. So along with the macrophages, we have T cells, B cells, and other immune cells together form a structure known as granuloma in the alveolar lumen. In the alveolar lumen. So these cells are held together or surrounded by a wall of fibrous tissue. and forms granuloma in the alveolar lumen. So this granuloma is for the actually for the protective purpose. It actually acts as a physical barrier to confine the bacteria to the specific place and prevent spreading. So it is actually for the granuloma is for the protective purpose so that the bacteria could not be able to spread to the other sides of the body. So this granuloma creates an environment where immune cells can interact with the bacteria and fix the bacteria at one particular location and prevent spreading. So the disadvantage will be the tissue damage in case of active tuberculosis. So this will be characterized by inflammation and destruction of surrounding tissues. So let me show you. So this is granuloma. So the granuloma, so these are the infected site. These are the mycobacterium. So these are the mycobacterium, and due to the infection of this is a specific site where the mycobacterium tuberculosis had invaded. So upon this infection, automatically we have the epithelioid histiocytes, which are activated macrophages. So this epithelioid lymphocytes comes together in order to form an association and enable themselves to bind with the mycobacterium family. So upon this, so epithelial histiocytes comes together like this and multinucleated giant cells. So the macrophages are giant cells, right? So the n number of macrophages along with some other T and B cells comes together form a multinucleated giant cells. So it actually look like a ball like structure. So this basically seems like a cheese ball. So this is a specific, this whole structure is known as a granuloma here. Granuloma. So this granuloma prevents the spread of infection. So when this particular granuloma ruptures, automatically the infected cells will comes out of the granuloma and via circulation they will infect the other parts of the body. So in order to prevent this condition, the immune cells comes together form the granuloma. So this is the structure of granuloma. I can show you here. Uh, so this is a macrophage antigen. So this is the, let's say this is the infected cell. This is the site of infection where we have the Mycobacterium tuberculosis. So with this we have the association of macrophages here. We have the association of macrophages and it seems like look at giant foam cell. This is whole the association of a macrophage where they actually bind with the antigens which are Mycobacterium tuberculosis. So this whole structure is known as granuloma. So in this cell count we have also have B cells. So in this association B cells we have B cells, T cells and neutrophils and mostly we have macrophages. So in the part of innate immune response or inflammatory cell recruitment and adaptive immune response. In as a part of inflammatory cell recruitment, we have T, B cells and neutrophils and also macrophages. So as a part of adaptive immune response, we have B cells are there Innate immune response, T cells, neutrophils are there. We have macrophages, which are high in number in the granuloma. So this is the whole granuloma structure. So what happens here? 90% in the 90% of the individuals, there will be the formation of granuloma in order to prevent the spread of infection in the body. In the 10% of individuals, there are chances of the spread of infection, which means this is the granuloma. Upon the rupture of this granuloma, there will be the spread of the Mycobacterium tuberculosis will comes out of the granuloma and spread to the other regions of the body, which cause active tuberculosis infection, which will happen in 10% of the individuals which uh, having weaker immune system. So this is the granuloma mediated protection to some extent, as well as the granuloma can also be the purpose of can also be the reason for the inflammation and tissue damage. So let's see the mechanism step by step. So how the mycobacterium? So even though the formation of granuloma for the protection purpose, the Mycobacterium tuberculosis will use the granuloma in order to spread itself to other parts of the body. So let's see how the Mycobacterium tuberculosis uses granuloma to its advantage. The first step will be the containment and protection. So, in order to reduce the active disease but also shield the bacteria from immune cells and other antibiotics. So, the granuloma actually forms in order to prevent the spread of infection. But here the granuloma is itself is like a ball, right? So, this granuloma hiding the infected cells from the hiding this infected cells or infected regions of the lung to the other immune cells, and also for the medications like antibiotics. So until unless the other immune cells could see this granuloma or could see this infected cells, they won't be able to act upon this infected cells, right? So this granuloma indirectly hiding this infected region to the other immune cells as well as antibiotics. So this will be the one disadvantage from granuloma. And the second one will be the modulation of immune response. Intracellular bactericidal mechanisms within the macrophages inhibit the functions of DC, dendritic cells, suppress the production of reactive oxygen and nitrogen species which are toxic to bacteria by modulating the activity of macrophages. So as there are presence of bulk number of immune cells, they actually couldn't able to act properly on the destruction of the mycobacterium. So eventually they start inhibiting the function of other immune cells like dendritic cells, also the suppress the production of reactive oxygen nitrogen species. So this species will be toxic to the bacteria. So this production will be suppressed by modulating the activity of macrophage. So this will be the one disadvantage. And the third one is survival and persistence. See, the formation of granuloma doesn't mean that the Mycobacterium tuberculosis have destructed in the region. It's not. So still, even though granuloma forms, the Mycobacterium tuberculosis can survive for decades as latent infection. So here the Mycobacterium tuberculosis had not destructed by the granuloma. It actually, the granuloma hides the infection and prevents the infection from spreading. So this Mycobacterium tuberculosis can survive in the granuloma for decades as latent infection, which means it is an inactive stage. It don't have another option to spread itself to the other regions of the body. That's why it was settled by the other immune cells to one specific location and exist as a latent infection. which means inactive stage, which means it can spread under immunosuppression. So whenever immunosuppression also, whenever there will be conditions like if the person had weaker immune system, have weak immune activity, the Mycobacterium tuberculosis can reactivate from the granuloma and cause disease. So as long as it is latent, there will be no problem. But if the immune system get weaker, if there are conditions like immunosuppression, there will be the reactivation of Mycobacterium tuberculosis, and there will be the chances of disease. So for this purpose also it used granuloma as vehicle for intracellular dissemination. So intracellular dissemination, which means the spread of disease, the spread of infected cells to the circulation and causing disease at other sides of the body. Here comes the necrosis. So in case of immunosuppression, so this is the case of immunosuppression where the mycobacterium use granuloma as vehicle. So upon necrosis, the mycobacterium will undergo reactivation. So necrosis is basically an unregulated cell death. So basically in our body, whenever there is a DNA damage, a cell damage, there is a program known as apoptosis. So apoptosis is the programmed cell death which will not cause any harm to the body, or which will not cause any tissue damage. So but in case of necrosis, it is an unregulated mechanism where it actually causes in the infected regions of the body infected cells, and eventually causes inflammation at that particular region. So, through necrosis, this granuloma undergoes necrosis by immune response or bacterial own mechanisms, and release bacteria leads to disease. So necrosis, I could show you the structure. So this is the granuloma, right? So upon during immunosuppression, this particular granuloma, so the particular infected cells will undergo necrosis, and there will be a rupture of the granuloma, and the infected cells or the mycobacterium will comes out of the granuloma and can spread to the entire body. So this mechanism will happen through necrosis, which means there will be a destruction of the entire tissue and the reactivation of the mycobacterium. So with this, release of bacteria leads to disease, and the macrophage and dendritic cells unable to present antigen to lymphocytes in granuloma thus leads to immunosuppression. See, all the cells together forms a clump, right? They could not be able to digest, engulf the antigen, and they could not be able to present the antigen to the lymphocytes as they are associated together at one specific location. They are not at all free. So they are all are in the duty of prevention of spreading of the infection. So there is no chance for the DC and macrophage cells to engulf and process it and antigen presentation to lymphocytes. So this become difficult, leads to an immunosuppressive conditions, leads to the spreading of the disease. The bacteria inside granuloma becomes dormant, which is an inactive stage, result in latent infection. So the symptoms here, symptoms will be coughing, a blood and mucus deep inside the lungs, weakness and fatigue, pain in chest, weight loss, and no appetite, chills and fever. So these are the symptoms of the person who are infected with tuberculosis. So how can we diagnose the tuberculosis infection? Through blood test. So TB blood test, also known as interferon gamma release. So through blood test we can diagnose it, and through skin test also. It is the most common type of diagnosis, where a protein known as tuberculin is injected at specific site, and if the swelling occurs more than 5 millimeters, then we can characterize the person is suffering with tuberculosis. And some imaging test, positive skin test like CT scan. So in the CT scan, if you observe the white spots in the lungs, it is also the presence of tuberculosis. In case of active TB, so white spots in the lungs shows active TB. As we can say, there will be the formation of granulomas in the lungs, and upon immunosuppression, the granuloma will be ruptured, and there will be a spread of infection which leads to active TB, right? So those granuloma can be characterized in CT scan. In some sputum test, if the chest x-ray shown sign like sputum or the mucus that comes up when you cough are tested in the sputum test. And some drug resistance 4 to 8, so some drug resistant to suggest medications, which means in sputum test they collect the samples of sputum, they test it with drugs, and after 4 to 8 weeks of the test results they will suggest medications to the patient. So finally, prevention and control, by BCG vaccine, Bacillus, Calmette, and green live vaccine. Through BCG vaccine, we can prevent tuberculosis, and this is BCG vaccine prepared from the strains of tuberculosis, Bacillus, and Mycobacterium bovis. So it is the only licensed vaccine against TB since 1921, and it is the most widely used vaccine worldwide. So this vaccine BCG will be 80% effective. So the second mode of prevention will be an healthy immune system. So 60% of adults with a healthy immune system can kill bacteria. So whatever the disease it may be, either it's a tuberculosis or any other kind of disease, if the immune system is weaker, then the person will prone to the infection severely. So if you maintain healthy immune system by diet or by our lifestyle, we can actually save ourselves from these kind of diseases. And early diagnosis, patient TB can infect 10 to 15 people per year, 2 weeks of medications. So this tuberculosis infection should be early diagnosed in order to prevent the infection from spreading. We should manage our environment, which means good ventilation must be there, natural light and good hygiene maintenance also prevents the disease. And treatment can be, if it is a latent TB, antibiotics are enough, sufficient, like isoniazid and rifampin. So these are for latent TB infection, and for active TB, ethambutol and isoniazid. So it is isonicotinic acid hydrazide basically, and some other like parazinamide. So these are some treatments for latent TB and active TB. So so this is all about tuberculosis infection, its mechanism of causing disease in the body. I hope you understand this lecture. Thank you.